Welcome back to Cancer Sequencing 101. Last time, we looked at how HiFi captures full-length isoforms and gene fusions in cancer. But today, we're going deeper into the epigenome. We'll cover two topics, DNA methylation and chromatin accessibility. Let's start with methylation. You probably know by now that cancer isn't driven by DNA mutations alone. There are also epigenetic changes, modifications that affect gene expression without altering the sequence itself, and they play a huge role. One of the most important is DNA methylation, specifically the addition of a methyl group to cytosine bases, 5-methylcytosine, or 5-MC. In cancer, aberrant methylation can silence tumor suppressor genes or activate oncogenes. In fact, promoter hypermethylation can act as a second hit, shutting down expression from the remaining allele after the first copy is already knocked out by a mutation. And traditionally, detecting methylation has required bisulfite conversion. That's a chemical treatment that's really harsh on DNA. It degrades your sample, destroys the long-range context, and it makes it almost impossible to phase methylation with nearby genetic variants. HiFi sequencing takes a completely different approach. It detects 5-MC directly from the native DNA. There's no chemical treatment needed. And here's how it works. As the polymerase reads each molecule during sequencing, the modified base acts as a molecular speed bump, subtly changing the polymerase's pace. HiFi measures these kinetic differences, and it calls the methylation status base by base. The calls get added right to your output BAM file alongside the sequencing data. So with every HiFi run, you're getting methylation information essentially for free. And because HiFi reads are long, typically 15 to 20 kilobases, you can phase that methylation with genetic variants on the same molecule. That's what makes this cancer example so powerful. In a sarcoma case, researchers found a 29 kilobase pair deletion on one copy of the tumor suppressor gene BRCA2. Short-read sequencing couldn't find a second hit on the other copy. But HiFi revealed the answer. The remaining allele was silenced by promoter hypermethylation. This is two completely different mechanisms. One, a structural deletion on a haplotype, and the other, epigenetic silencing, working together to knock out a critical tumor suppressor. You can only see that when you have long reads that phase genetics and epigenetics all together. Now let's look at chromatin accessibility. Chromatin architecture, or how tightly DNA is packaged around histone proteins, controls for which genes are accessible to transcribe. Changes in chromatin accessibility are key drivers of cancer development. This is where the Fiber-seq assay comes in. Here's how it works. First, you take your cell nuclei and incubate them briefly with an enzyme called HiA5. This enzyme adds a different methylation mark, 6-methyladenine, or 6-MA. But it only adds it to regions where the chromatin is open and accessible. It's essentially stenciling the chromatin state directly onto the DNA. So then, when you sequence with HiFi, which natively reads both the 6-MA accessibility marks and the 5-MC methylation along with the DNA sequence, the result is that HiFi simultaneously reads all 4 bases plus 5-MC and 6-MA, giving you the genetic and epigenetic variation from a single molecule. That means you'll get the genome sequence, CpG methylation, chromatin accessibility, and even transcription factor footprints all in one assay. Here's a striking example. Researchers used Fiber-seq to investigate a case of bilateral retinoblastoma. They discovered a translocation between chromosomes 13 and X that placed the RB1 tumor suppressor gene onto the X chromosome. In about 3% of cells, that fusion chromosome underwent X inactivation, silencing RB1 through a chromatin accessibility change, not a mutation. That's the kind of mechanistic insight that's only possible when you can read genetics and epigenetics on the same molecule. To explore more cancer biomarkers revealed by HiFi, visit our interactive cancer body map. The link is in the description. And next up, we're looking at how HiFi resolves the structural architecture of cancer genomes. Thanks for joining me.